Good evening. Welcome to the Museum of Science. My name is Insoo Hyun, and I'm the director of the Center for Life Sciences and Public Learning. Here, the purpose of my center is to leverage the enormous life science talent in the communities around us and to bring cutting-edge content through the museum out to diverse audiences to just ensure that no one is left behind as new scientific developments push our world forward. In the year 2025, the museum will be shining a spotlight for the entire year on the year of being human. The theme is the science of who we are and how we thrive. And look forward to programming all year a lot like this, which celebrates scientific discoveries and connections about us and the natural world, and just really excited about that upcoming spotlight. Now tonight, the museum is honored to partner with the National Academy of Sciences to host this event, a special evening of science and conversation celebrating Dr. Feng Zhang's groundbreaking achievements and his current work that explores how biological diversity can help us develop new CRISPR systems. The Richard Lounsbery Award is given by the National Academy of Sciences and the French Académie of Sciences to recognize extraordinary work in science, in biology, and medicine. In addition to honoring scientific excellence, this award is intended to stimulate research and encourage scientific exchanges between the US and France. Tonight's event is reaching a very broad audience, both here in person and through a robust live stream online. Now, I should note that this is the first time this award lecture is being given in a place other than a traditional academic institution. So in the spirit of bringing current science to the world, it is important that you, the audience, also participate by submitting your questions to Dr. Zhang by going to slido.com. And at that website, you can enter the code Lounsbery, all one word, Lounsbery, and submit your questions during the program, and we'll get to as many of those as possible during our conversation after the lecture. Now with that, I would like to introduce our museum president, Tim Ritchie, to come up to the podium and say a few words. Tim.
Well, thank you, Insoo. I want to say just a little bit more why we're so glad to have the Richard Lounsbery lecture here tonight. And in a word, this is what we're becoming. I don't know if you've noticed, but our entire second floor practically is under construction. And in 2 years from now, we will build the finest public convening space in the world. And we will have more such lectures like this because it's our goal to gather the public, to gather industry, to gather academia, to gather government together to think out loud about what kind of world we want. How do we use science and technology to build a future that is humane and sustainable? And so that's why we're so thrilled to have this here. And it's here because the recipient of the prize, Feng Zhang, asked for it to be here. And I think that that's just a really remarkable thing because of this concern about public engagement with science, not just an academic gaze. Of course, if it were just an academic gaze, still Feng Zhang would be the one we would be honoring. I'm going to tell you a few things about him. Many of you will know this, but perhaps not all. Dr. Zhang is a core member of the Broad Institute of MIT and Harvard, where his groundbreaking work has significantly advanced the understanding of genetics. He holds the prestigious James and Patricia Porteous Professorship in Neuroscience at the McGovern Institute for Brain Research at MIT, and he's also an investigator at the Howard Hughes Medical Institute. His contributions to the development of CRISPR gene editing technology have opened new avenues in biological research and therapeutics. His innovation does not stop at CRISPR, however. It extends also to the instrumental work he's done with optogenetics, which is a technology that uses light to control cells within living tissue. Throughout his career, he's received a number of awards, including the Gardner Foundation International Award and the Tong Prize in Biopharmaceutical Science for his role in developing CRISPR-Cas9. His work's not only, of course, a testament to scientific excellence, but also to his own dedication to solving some of the most pressing challenges in human health. When you ask people about Feng Zhang, what they say most is they comment on most, is his contagious enthusiasm for science and his genuine support for his peers and for mentees. They say his approach to science is meticulous and innovative, and yet he remains humble and approachable, characteristics that truly set him apart as a researcher and as a mentor. What I want to say are two things that I'll bet you don't know. One is his name. His name actually means edge of a tool or point of a spear. How appropriate for someone who's one of the world's great creators of tools that cut things. So I thought that was, and then the second thing is that Dr. Zhang, Feng Zhang, grew up in Des Moines, Iowa. And he grew up as a boy going to the Iowa Science Center. And that did change his life. That's what science centers do. We change lives. And I'm so glad you went to that institution and that you're devoted to this institution, your local science center, the Museum of Science. So for that, please join me in welcoming Dr. Feng Zhang, a visionary scientist whose contributions continue to inspire science and propel people to do great things in the world. Dr. Zhang.
Great. Good evening. Tim, thank you so much for the kind introduction. I want to start by thanking the Museum of Science and also the Lounsbery Foundation and also the National Academy of Science for making this opportunity possible. And I'm also really excited to be here because, as Tim mentioned, I grew up in Des Moines, Iowa, and going to Science Center. And nowadays I have young kids, and they love coming here. And this is really just a wonderful community and wonderful environment to really inspire and also to be curious and to learn things. So I put together a few slides for tonight. I thought I'll tell you a little bit about some of the work we have been working on. And also I'm looking forward to engaging with everyone and talking more science. So I grew up in Des Moines, Iowa. And I kind of got excited and stimulated about biology when I was in middle school. So in 7th grade, I went to a Saturday enrichment class where they showed us this documentary. And it was a class on molecular biology. And I really didn't like biology before that because 6th grade biology or life science was very much about memorization. Dissecting frogs and memorizing anatomical parts or remembering shapes of different leaves, it didn't sort of excite me as much. But when going to this class where they began to teach us about the molecular biology principles, how things work in central dogma, where DNA becomes RNA, and then RNA is used to produce protein inside of the cell, that made it feel like biology is something that has first principles, and you can start to engineer it based on those first principles. So that kind of got me excited. And of course, watching this documentary and seeing how people can create dinosaurs made it even more exciting. So Tim mentioned about mentorship, and this is something that also had a profound impact on my life, which is that the teacher who taught this class was one of the science teachers in my public school. And he remembered that I, along with several other students in the class, were very excited about molecular biology. And so by the time we went to high school in 10th grade, this teacher came to us and he said that in the local hospital, the Iowa Methodist Medical Center, they just opened a new gene therapy lab. And the hospital takes volunteers so you can go and intern in different departments to learn things. And he suggested that maybe we apply into the internship program and then specify that we wanted to work in the gene therapy lab. He also caveated by saying that you might not do anything sophisticated, you might just be washing glassware, but it's worth trying. So we tried and we got in, and then we began to start to learn what real scientific research in biology is like. And so I was paired up with a staff scientist there. His name's Dr. John Levy. And then working with John, I began to work with cells, work with DNA, and I still remember the very first experiment he had me do, which was to put the gene for a protein from a jellyfish. It's called a green fluorescent protein. So we had human cancer cells, and he asked me to put a drop of clear liquid containing this jellyfish gene onto those cancer cells. And the following day, he took me, along with that petri dish of cells, into a dark room with a microscope. And the microscope had a beam of blue light coming through the lens. And he said, "Feng, look into the objective of this microscope and see what you see under this microscope." And I remember being completely overblown by what I saw under the objective. The cancer cells were glowing green. And I had never seen something that's so beautiful and so amazing before. And John said, "Remember this little drop of clear liquid that you put on yesterday? That contained the gene, and that gene was used to make this fluorescent protein that is normally in a jellyfish, but now you transferred it into human cells." So that was my first experiment of doing gene transfer. But that really inspired me to continue to work on biology, because the promise of being able to use genes to be able to understand human and also to be able to treat disease was just such a profound and impactful concept. So since then, I have been working on this area called genetic medicine. And so I put over here a picture of a rocket delivering a satellite into outer space. And this is kind of a rough analogy of how genetic medicine works. There is a vehicle, and then there is a payload. So the vehicle, usually in the form of either chemicals or in the form of a little virus, can deliver a genetic therapy payload into the right cells to either kill cancer cells or to be able to fix eye cell, retinal cells in the eye, or brain cells or liver cells, to be able to treat disease. So this modular concept is very powerful because it suggests that if we develop a toolbox of different vehicles, different, something that goes into the brain, or something that goes to the eye, or something that goes to cells in the blood, and we also develop a toolbox of different payloads, something that can turn genes on or something that can turn genes off, something that can change the state of a cell. So by mix and matching the right vehicle with the right payload, you can have a modular way of developing genetic medicine. So you can treat diseases in the eye, diseases in the brain, and so forth. So that's kind of the underlying concept of genetic medicine, and we have been working on it. And there are many brilliant scientists in the field that are now making really exciting progress to treat disease. So what has really fueled this development, one of the most important things is the sequencing of the human genome. So here is a picture showing all of the different chromosomes in the human genome. And so more than 20 years ago, the first human genome sequence was completed. And so the human genome has 3 billion letters. And so each one of us has 3 billion letters, and we differ from each other slightly. And now, the first human genome costed almost $1 billion to complete. And now people can sequence whole human genomes for maybe a couple $100. And so with that significant drop in the cost, scientists and doctors have been able to sequence many people's genomes. In fact, hundreds of thousands of human genomes. By comparing the letters in these genomes, scientists have now identified more than 5,000 diseases that are caused by genetic differences. Things like sickle cell disease, or cystic fibrosis, or degeneration of the eye, or Huntington disease. These are all what's called genetic diseases that are caused by specific mutations. Now that we know what causes genetic disease, one of the tantalizing ideas is, what if we can go into the cells and be able to fix them? Replace the disease-causing mutations with something that is found in healthy individuals. So this is where the idea of gene editing comes in. So it turns out that gene editing, or this idea of trying to fix DNA sequence, has been worked on for many decades, and multiple iterations of technologies have been developed. The latest generation of technology is something that, if you had a bowl of yogurt this morning, you have had it. And this is something called CRISPR. So CRISPR is a system that's used in bacterial cells to be able to fight against viruses that infect bacteria. And people in the yogurt industry use this natural process to be able to immunize bacteria that people use to make yogurt to become immune to viruses, so that if they're fermenting a large, say 100,000 liter vat of dairy product, a virus is not going to go in and spoil it. So this is used 100% in yogurt production. So if you had a bowl of yogurt, you have had CRISPR. So this is a picture of a bacterial cell, and these little things that you see on the top, these are bacterial viruses. And so just like us, we can get infected by the flu, by COVID, by all sorts of RSV viruses and so forth. Bacteria also can get infected by all sorts of viruses. And so CRISPR is a way that these bacteria have evolved to be able to defend themselves against these viruses. And so the way it does it is by snipping out a little bit of that virus, so something that is kind of a signature for that virus. It will remember it, and the next time when the virus comes in, that little bit of signature is used by the bacterial cell to recognize that a virus has come in, and then initiate a whole cascade of destruction mechanisms to get rid of that virus. So that's what CRISPR does. So we and also others have worked on CRISPR, and we have harnessed it to be able to use it as a tool to recognize mutations in the human genome and to be able to fix it. And so these are two of the ways that people use CRISPR. They either use this protein called Cas9 from CRISPR to be able to go in and recognize DNA sequences in the genome and to be able to modify it, or they use it to be able to recognize a specific spot in the genome and then bring other machineries over to be able to turn things on or to be able to turn things off. And so this has become a very powerful way that is used in almost many biological laboratories to both study biology, to understand more, and also to be able to develop treatments for disease. So one of the really exciting aspects of CRISPR is that it uses a short RNA to be able to recognize DNA. And this RNA is something that can base pair with DNA. So DNA has 4 letters, A, G, T, and C. And RNA has 4 letters, A, U, T, and C. And so you can design RNA that matches the DNA to then be able to recognize sequences in the genome. And so what we can do now in the laboratory is that we can use chemical synthesis to make tens or hundreds of thousands of RNA sequences that can recognize almost every single gene in the human genome. And then we can carry out very large-scale experiments to ask questions like, which one out of the 20,000 genes in the human genome is involved in causing cancer? Or which one of the 20,000 genes in the human genome is involved in allowing a cancer cell to become resistant to the cancer therapeutic? And so this is something that's called genetic screening. And it is possible now to do that because it's very easy to use chemical synthesis to make these kinds of RNA at very large scale. So we have applied this method to study disease and also to be able to treat disease. And so the way that people go about treating disease using gene editing comes in 2 different forms. The first form is called ex vivo gene therapy. The way this works is that people would take cells out of the patient's body fix those cells, remove the mutation, and then be able to put the cells back into the person. This works very well for cells in the blood. People have been doing bone marrow transplantation for a long time now, and people have also developed T-cell, immune cell therapy for treating cancer. And so with gene editing, what people can do is they can take bone marrows out of the patient, fix those bone marrow cells, and then put it back into the patient so that the person can become cured of that disease. This works for cells in the blood or cells in the bone marrow, but it doesn't work for other cells. We don't have yet the technology to remove someone's brain, keep it alive in a petri dish, fix it, and then transplant it back into the person. So in order to treat diseases in those other organs, scientists are working on in vivo gene therapy. This involves injecting the medicine, whether it's gene editing medicine or gene therapy medicine, directly into the body. This medicine can be modified to target specific tissues. For example, if you wanted to go into the brain, you can try to modify it so that it crosses the blood-brain barrier and can get into brain cells. If you wanted to get into the liver, you can design it so that it goes into hepatocytes in the liver. So those are cells in the liver. So these are things that the scientists are now working on to be able to treat diseases in different organs without having to take the cells out of the patient. So both of these approaches are now actively being pursued in the clinic for development. There are clinical trials that are using gene editing to go after cells in the blood, like sickle cell disease, or people are using it to treat specific organs in the body, like liver diseases, or to try to be able to develop treatments for Huntington's disease in the brain, and also many others. So one of the things that has really advanced quite significantly was the treatment of sickle cell disease. And so just about a year ago, both in the United Kingdom and also in the United States, a new gene editing therapy for treating sickle cell was approved called Casgevy. And so this really built on a long cascade of work. And so one of the major discoveries was based on Stuart Orkin's discovery that there's a gene called BCL11A. And if you can turn this gene off, then you can get a sickle cell, a sickled red blood cell, to become unsickled, because it can then turn on a form of hemoglobin that is only turned on in infants. So by turning on this fetal hemoglobin, then you can make the cell healthy again. So then, knowing this, one of the questions is, how do you go and turn on these fetal hemoglobin genes? Or how do you turn off this gene called BCL11A so that fetal hemoglobin can get turned on? And so we worked with Stuart Orkin and also Dan Bauer to carry out a genetic screen. We designed these CRISPR systems and RNA to be able to cover every single spot along the regulatory region for this gene called BCL11A. And CRISPR would go in and allow us to make one mutation at a time, throughout all of the different spots covering this regulatory part. And we found that there's one specific guide, one specific RNA called sgRNA 1617. It can go in and make a very specific modification in the part of the genome that regulates BCL11A, turn it off so that fetal hemoglobin can get turned on. So it's using this strategy that Vertex Pharmaceuticals and CRISPR Therapeutics, they put this into human cells. They took bone marrow out of patients, and they modified BCL11A using this strategy, and then they transplanted the modified bone marrow cells back into the patient. So the picture on the right shows you the first patient who was treated using this strategy, and her name is Victoria Gray. And so after receiving this therapy, her sickle cell condition was cured. She is no longer dependent on going to the hospital to get blood transfusions, and her sort of bone marrow cells have been fixed so that fetal hemoglobin gets turned on in her blood cells. And so a number of patients have been treated with this therapy now. And so last year, the FDA in the US and also the regulatory agency in the UK approved this first drug called Casgevy. And this is a whole process of taking cells out of the person, fixing the bone marrow cells using CRISPR, and then putting the bone marrow cells back into the patient so that all of the new cells that are produced are no longer sickle. So this is just the tip of the iceberg. There are many, many other therapies that are now under development. There are multiple biotechnology companies that are now developing CRISPR as a form of medicine to treat diseases in the blood, to treat cancer, to treat liver diseases, eye diseases, and even muscular and also neurological diseases. So a lot of these clinical trials are actively underway, and hopefully in the coming years, we're going to see more and more drugs being approved where we can go in and start to cure diseases rather than just masking the symptom. So CRISPR has come a long way. But if you look at clinical trials that are getting developed, they largely focus on three tissues. That's cells in the blood, cells in the liver, and also cells in the eye. And you're probably wondering, we have a lot of other tissue in the body. We have skin, we have kidney, we have pancreas, we have the brain. Why aren't there more clinical trials targeting these other tissues using gene editing and gene therapy? The reason for that is that the delivery system, the ways that we can get the drugs into those tissues have lagged behind. We can get into blood, we can get into liver, we can get into eye relatively effectively, but these other tissues, we still need to develop new methodologies. And so to make gene editing more effective and to sort of unlock some of these other tissues, we have been working on developing new delivery technologies to be able to access these other parts of the body. And so I put together a few slides on two short stories that we've been working on in the lab. And so the first one looks at using some of the proteins in our own body to be able to deliver things into different tissues. And then the second story looks at harnessing something from bacteria and using those things to be able to deliver gene therapy into human cells. So let's talk about the first one. So the human genome was sequenced more than 20 years ago. And there are 3 billion letters, and there are 20,000 genes that are encoded by the 3 billion letters. When that was done, scientists began to start to analyze these 20,000 genes. And so one line of question was that how many of these genes evolved from viruses that bombarded our ancestors? So what they did is that the computational scientists took virus genes, real virus genes, and they searched along the 20,000 genes in the human genome. And they found that some, about 80 human genes, share very strong resemblance to these viral genes. And so that suggests that over the course of 50 to 100 years of, a million years of evolution, our ancestors were infected by what is called retroviruses. These retroviruses leave copies of themselves in the genome of our ancestors. And so as our ancestors reproduced, and generation after generation, those viral elements got retained and got passed on to our present day selves. And so some of these proteins may have preserved their function as virus-like things. Viruses that are really good at infecting cells and delivering their genetic information to the cell, and then replicating, and then leaving, and then infecting more cells. This is a natural way of delivering things into cells. So we thought, let's study these human genes to see what they may do. And then about 6 years ago, there were two really exciting studies. One from Jason Shepard's group at the University of Utah, and another one from Travis Thomason's group at the University of Massachusetts. And they studied one of these genes called ARC. And they found that the ARC protein is very similar to virus proteins, and they form virus-like structures even in animal cells. And so we thought this is really cool. So maybe we can start to study these more. So 3 of my lab members, Vicky, Yu Gang, and also Ramya, they began to look at one of these groups of proteins. They're called PNMA, and these are genes that are found in the human genome. Not very much is known about these genes, and so we thought to study them and see what they do. They studied all of them, and they found that they all can form virus-like structures. And then one of them, called PNMA2, so number 2 out of this family, forms virus-like structures that look very much like a form of virus that infects human and is used in gene therapy called AAV, adeno-associated virus. So if you look here on the slide, you will see these little particles. This is an image taken by an electron microscope. So they're very, very tiny, about 20 to 30 nanometers in diameter. So very, very, much thinner than even the diameter of a piece of hair. And these things are what viruses look like. And so we thought this is really cool. Maybe let's study them and see if we can harness them to be able to deliver useful things into cells. We first tried to see what do they package, and this is an experiment we did where we found almost nothing. So if something was packaged, it would be above this red dashed line. And we found that nothing really got packaged. So these were empty particles. That's okay. Let's figure out what they look like so that we can see if we can engineer them to be able to package material. So we studied that using bacteria. And what we did is that we used E. coli to make lots and lots of this human protein. And this is something that's very easily done now. And what we found is that bacteria can make them. So in fact, if you look under the microscope, they form virus-like structures even in bacterial cells. And these are very easy to work with. We can grow lots of bacteria will make lots of the protein, and then we can purify them. And they behave really well. So they are almost like a Lego system. If you have these proteins in a tube of liquid, in a buffer, if you remove the salt from the buffer, these virus-like structures will fall apart. So if you remove the salt, if you put in 0 amount of sodium, salt, all these structures will break open. They'll disassemble. And if you put the salt back in, so this is half a molar of sodium chloride, table salt, then they will self-assemble back into these virus-like structures, just like these. So this is really cool. So now we have a virus. We can remove salt to open it up, and then we can add salt to get it to come back together. So it can fall apart and piece it together. So it's a nice way to open it up and then close. So we thought if we can open it up, maybe we can put something into it. But then we had to figure out how to put something in. Here is a much higher resolution of image of what these virus-like things look like. This is done using cryo, so freezing electron microscope. And this is a cross-section of this virus-like structure. So you're looking at the inside of this virus. Red means negative charge, and then blue means positive charge. So what you see is that the inside surface of this sort of cut open virus is negatively charged. So we want to package DNA or RNA into this. DNA and RNA naturally have has a negative charge. And so if the inside is negative and DNA and RNA are also negative, they're going to repel each other, right? It's like 2 different pieces of magnet. If you put the same pole together, they're going to repel. So what we had to figure out is how do we change the inside so that it's positively charged, so that it will attract a negatively charged DNA or RNA. So we did that by engineering the protein. So we took the natural version of PNMA2. We cut off this little region at the end of the protein, and we replaced it with something that is positively charged. And this is a piece of protein from a plant virus that is known to be able to interact with the on the inside. You can use these kind of physical and chemical principles to really engineer the system to get it to work the way you want it to work. So that's very cool. So we then used it to package a gene. we use it to package something called recombinase, so that if we put it into cells, it will turn the cell green. And so these are what we found. So if you use these engineered virus-like PNMA2s to deliver recombinase to the cells, you can make the cells turn on the reporter and glow green. And this is exactly what my very first experiment in the high school gene therapy lab looked like. These are the cells that you would see under the microscope where they grow this amber-like color, very, very beautiful. So this is something that they're working on now. What is very exciting is that these are human proteins, which means that if you use it and put it back into the human body, they are probably going to be much better tolerated than something that didn't exist in the human body before. Our immune system will tolerate it better. And so this is something that we're quite excited to be working on. So then the second approach that we have been working on is to harness something from bacteria. So Joe, who is a graduate student in my lab, has been working on harnessing something from bacteria that's almost like a nano syringe that can directly shove things into cells. And this is what they look like under electron microscopy. So this is a high-resolution image of what these syringes look like. You can see they are very long. They have a sharp spike at the tip. And then they also have a hollow core on the inside where you can actually load things into it. This exists in nature. So nature invented it, created it, and bacterial cells use this to be able to fight each other by injecting chemical signals back and forth to either kill and suppress other cells or to allow other cells to grow even better. So it's a way for them to deliver information between each other. And this is how the injection mechanism works. This is a schematic. So the syringe naturally looks like this, and the syringe has these gray things that are called tail fibers. They're almost like the landing gear that a lunar landing vehicle would have to land onto the surface of the moon. And so they have ways to be able to recognize in a specific way specific types of cells. And so these things use the landing gear to attach on the cell surface to then be able to recognize it. Upon landing, this spring-loaded mechanism will get activated. It will compress, and then that compression will shove this spike protein, push it through the cell membrane to then deliver a payload in the recipient cell. This is how this system works. It's very, very cool. And so we started to work with this system. We borrowed it from a pathogen called Photorhabdis, and we transferred just the components for making more syringes into E. coli. So we were then able to use E. coli to produce lots and lots of these syringes. So you can see they're long and narrow, and they all have things loaded inside that can then go in and inject into other cells. And if you look very closely, each one of these has a little sharp tip at the end, and that's how these things inject into cells. So we had to do some engineering. So there's an AI exhibit upstairs, and we used AI tools to help us engineer the system. So these landing gears that allow this syringe to attach onto cells normally only recognize cells in insects. But we want to get this to recognize human cells. And so the way we did this is we used the AI program called AlphaFold 2 to be able to predict what these landing gears look like. And so this is what the AI program predicted that the protein would look like. Once we know what the predicted shape of this protein looks like, we were then able to engineer it. So we can reason to make a cut here to get rid of this part that touches and recognizes the recipient cell. And we can replace it with something else that can recognize human cells. So this is a different protein from a virus that can recognize human cells. Or we can replace it with this domain that is able to recognize a protein on the surface of a cancer cell. So these are the different forms that we engineered. And so we again use this reporter system to see whether or not it would work. So we have a cell, if it can receive the payload, it will turn green. With the wild type version, so with the natural version, there's no reporter, no greenness in the cell. So because it didn't recognize the human cell. And then if we cut off this landing gear attachment domain, there's also nothing. But if we put on this virus domain that can recognize human cells, we can now see greenness in the cell. So the reporter gets turned on. And then if we mutate it so that it doesn't recognize human cell anymore, there's no signal. And then if we put on this domain that can recognize the protein on the surface of this cancer cell, then we can also get a signal turned on. And so this suggests that we can also use rational engineering to be able to change this injection system so that rather than injecting into an insect cell, you can inject into a human cell. So then we show that you can use this to inject gene editing proteins into cells to be able to do something more useful. And so this is using either the CRISPR Cas9 protein or this is using a zinc finger deaminase, two different forms of gene editors. You can use this to then achieve gene editing in the recipient cell. And also we can use this in a very modular way. So the domain that we attach onto this landing gear, we can attach different types of domains. And they have specificity. So only when the domain matches with what is on the surface of the recipient cell do you get successful injection. And so this is what you see here. So across the diagonal means that, so over on the x-axis are different domains on the landing gear, and then across the y-axis are different proteins on the cell surface. And so only when the domain that's on the syringe matches the protein that's on the cell surface do you get signal. If this doesn't match, then there's no signal. And so this showed us that we can quite successfully retarget this syringe to go after different types of cells. And so that makes the system quite modular. So anyway, so that's what we're working on. We're both exploring these human proteins as well as bacterial proteins, making them as modular as possible so that we can have a broad toolbox to be able to develop, to deliver genetic medicine to a variety of different types of cells, tissues, and to treat diseases in all those different tissues. So one of the concepts that's underlying all of the things that I mentioned today, whether it's CRISPR, or is these human PNMAs, or is the bacterial syringes, they came from natural diversity. So they were things that nature evolved over tens to hundreds of millions of years. And so it's really important to then explore this natural diversity. And so the plot that I'm showing you here on the right side shows how rapidly we are collecting information from this natural diversity. There are bacteria that live on our skin, in our intestines. There are bacteria living on the surface of this podium. There are bacteria living on the escalator, on the red line T, everywhere. And there are gazillion different species of bacteria. And we need to sequence them. We need to collect their genetic information and collect and build as large of a repository as possible so that we can go in and mine all of the richness that nature has created. And from there, we can learn a lot about biology, but also we can develop new and powerful transformative tools to be able to improve human health, solve climate change, and to overall make the world a better place. So that's what we're working on. We're excited to continue this. And then last but not least, I just want to thank again to the Lounsbery Foundation, the National Academy of Sciences, the Museum of Science of Boston for hosting this phenomenal event. And it's really a great pleasure and honor to be here to interact with all of you and to share our science. And then finally, this is my team at MIT. And also we're supported by a number of different funding sources to allow us to carry out this work. Thank you again, and it's really a great pleasure and honor to be here.
Thank you. Thank you so much for that wonderful talk. I want to remind the audience and the folks online to submit questions going to slido.com and enter the code word Lounsbery. So as those questions are coming in, I'll just start off with a few questions of my own. I'm going to start with where you left off about biodiversity. There are people who are worried about the loss of biodiversity of large animals and birds for very good reasons. Is there also a threat of loss of biodiversity in the micro world? And is that something that worries you?
I think it's definitely very important to collect as much information, to save as much biodiversity information as possible. The world is changing, climate is changing. This is middle of November in Boston, and we can still walk around outside and not be freezing. And so a lot of species are coping with these changes, and so animals, even microbes, are disappearing because of these changes. So what is really important to do, and a lot of scientists are working on now, is to use DNA sequencing to go into all sorts of ecological environments, collecting biological samples, and then sequencing the genetic information, and then forming a large repository of that information to then keep a record of what has existed. That information can tell us about what are the genes, what are the proteins, what are the molecular processes that existed in these organisms. And by looking into these mechanisms, we can harness things that are potentially useful. So for example, there may be bacteria that live in the Amazon rainforest that have some magical compound that's able to kill cancer cells. Or there may be an organism that has a machinery, a protein, that can degrade plastic that we can use to be able to mitigate plastic waste. And if we didn't sequence and keep a record of this biological diversity, we would have lost all that biochemical sort of tool that nature has evolved. So I think it's incredibly important.
That's so interesting. So earlier in the year, we had Craig Venter here in a conversation like this in front of an audience, and he was talking about his work sequencing the ocean's microbiome. And so you're saying then that kind of work, that kind of data is incredibly useful for the kinds of applications you're thinking of.
Yeah, sequencing any biological diversity, and the ocean has a large amount of diversity that we can get information from. So things that are, the ocean is large. There are things that live at the surface. There are also things that live deep under the ocean. There are geothermal vents, deep on the bottom floor of the ocean, and these diverse environments allow or cause bacteria to evolve very different ways to survive. And I think by mining this diversity, we're going to be able to find a lot of cool and powerful tools.
It's not lost on me that your speech is happening right next to some capsules that we have, space capsules. And I thought it was so fascinating. So much of our work now is devoted on the vehicle by which you're delivering these editing systems. When you were describing the syringes and making modifications to the landing gear, that syringe, what tools are you using to make those cuts or those changes?
We use a lot of different tools from molecular biology to be able to engineer it. So we use a technique called recombinant DNA. So things like polymerase chain reaction so that we can copy different pieces of DNA. Ligation to be able to join different pieces of DNA together. We use bacteria to be able to amplify, make many, many more copies of it. So those are all of the basic tools in the molecular biologist toolbox. And we use that, and many people use it to be able to do these experiments.
Okay. Now, I know questions must have come in, but I might not be on the right page. So if James is here, maybe you can help me get the... but in the meantime, let me ask one other question. So there are other gene editing tools historically, and Talons, I believe, was one that plants have used to sort of fight off viral attacks. Why has that kind of gone a little bit out of favor, and why has CRISPR sort of taken over? Is it because you have a longer guide RNA, it's more useful? I'm curious about that switch to CRISPR.
Sure. So gene editing technology has evolved over multiple generations, and the direction that they were getting optimized on is how easily can we reprogram it to recognize a new gene in the genome. So the earliest technologies like meganucleases or zinc fingers were incredibly hard to be able to reprogram. And then talents came along, which was a little easier, but still cumbersome. So those things probably take at least a week, if not longer, to be able to make something that can recognize a new gene. And sometimes, even when you go through all that work, it may not work very well. When CRISPR came along, the amount of time that is required to make something dropped to just a matter of 10 minutes. So you can just literally go onto the computer, type in a sequence, and then a couple of days later, FedEx will send you an envelope with a tube of RNA or DNA that you designed. So is that simplification of the design process that made CRISPR so much more attractive?
Well, that's fascinating. Okay, so we do have a bunch of questions. One question, you mentioned about how the cost of sequencing the human genome has come down significantly. Do you foresee off-the-shelf gene therapy products in the future that can be more affordable, widely available to patients who need it? How would you even go about developing such technologies? This is about the cost.
Yeah, that's a good question. In some ways, gene therapy has already reached massive scale. And it's kind of off the shelf in the sense that you can walk into Walgreens or CVS and get it. And that is the COVID vaccine. So COVID vaccine is a form of gene therapy. It's an mRNA that is packaged by something called the lipid nanoparticle. You walk in and you can get a jab, and billions of people around the world have gotten it. So that is one form that is probably the most advanced. And I think in the future, certainly we're going to see more. There are going to be more RNA vaccines for other diseases besides COVID, and we're going to see mRNA or gene therapy as a treatment for other diseases too. For treating really complicated diseases like Alzheimer's disease or cancer, I think we don't know enough yet to develop successful gene therapy, but ultimately we're going to know enough, and then we'll probably have medicine for treating them.
There's another very interesting question that's come in. In the U.S., kidney disease continues to be a problem, seems to be increasing. Do you have any insights on the path for developing a gene therapy for kidney disease? And let me add to this. I mean, clearly, it's hard to do in vivo gene therapy or delivery. There are some people, maybe even here at Mass General, who are working on removing organs from people, biopreserving them for a long period of time outside the body, maybe to do something like gene therapy. So maybe that might be one path for that, but some thoughts on the path to treating kidney disease.
There are a lot of people who are working on trying to develop delivery systems for the kidney. So the reason that gene therapy is hard in the kidney is because it's hard to get things into kidney cells. Part of the reason is because we have so these organs are tissue, and tissue are made up of many, many cells. And so between the cells, there is a little gap. And that gap varies depending on which tissue you're working in. In the kidney, that gap is very, very tight. And so if you are trying to deliver gene therapy into the body, the gene therapy has to be able to diffuse throughout the tissue to get into those cells. And some of these gene therapy particles are larger than the gap between those cells. And so that means they will get clogged, they will get stuck. And so new ways to be able to get things to distribute in these tissues need to get developed. And it's challenging, but I think people are making progress there.
So along the lines of different types of treatments, tell us more about gene therapy for eye diseases. What kind of diseases are we talking about here? How are they being treated?
There are eye diseases that are being treated using gene therapy, usually using viral vectors. So these are natural viruses. They either are pathogenic or they can be non-pathogenic. Scientists take these viruses and they remove all of the genetic information from that virus, and they replace it with the genetic information for the gene therapy drug. so that they're basically using the shell of the virus to deliver drugs into cells. And so there are mutations in the eye that cause blindness. So for example, LCA2 is a disease that is affected in some individuals. And so people have developed AAV, or virus gene therapy, for treating these blindness. And so one of these has been approved by the FDA several years ago. And so there's a company in Philadelphia called Spark Therapeutics. They developed the very first gene therapy drug for treating blindness, and that was approved for treatment. So you would take that virus vector, inject it into the retina, and then once it's injected there, it will diffuse and be able to infect retinal cells to then be able to recover, to allow the cells to be able to sense light again. So that's one form of treatment. And there are now people exploring using these viral vectors to treat other forms of blindness or treat glaucoma or other diseases affecting the eye.
Another question about treatment. For sickle cell disease, when it's treated in the way that you described tonight, is that benefit passed on to offspring, or would the offspring have to also undergo the treatment? Why or why not?
The gene therapies that I mentioned so far, none of those will pass on to the offspring, because the gene therapy, they don't modify the germ cells in the body. So germ cells are the reproductive cells, the sperms and egg cells. So because we're not modifying those cells, the offsprings will not receive the modification. So we're only changing cells in the body that are called somatic cells. And when it comes to changing germ cells, there are a lot of ethical challenges involved there. And so I don't know, you might remember that back in 2018, there was a scientist who used gene editing to modify the two IVF embryos. And then those embryos were then reimplanted and sort of led to the birth of 2 baby girls in China. And that caused a huge international sort of uproar. And so germline modification is something that is very much sort of very hotly sort of debated about whether or not any form of that should be conducted right now.
Right. In 2017, I believe the National Academy of Sciences produced a report on the science and ethics of genome editing, and this is really a hot-button issue that they had in terms of the criteria one would have to meet to actually justify going in that direction. So we have time for one more question, and I'll just end with a question I have for you. You, as a child, went to the Science Museum in Iowa, and your children come here to the Museum of Science and enjoy the time here. What kind of future do you hope you'll leave your children in terms of gene therapy and these discoveries that you're working on? What are your hopes for that generation?
I have a lot of hopes for the next generation. I hope the next generation will continue to be curious, continue to to pursue science and follow science and discovery, and use those things to solve the most important pressing problems of their time, to make the world a better place, make people's lives better, and also be responsible for the environment that we live in, so that we are not only making the planet a good place for our species, but also for all of the other species that we share this planet with. We have a responsibility for doing that.
Well, you're getting us off to a very good start. We're so lucky to have you, not only here at the Museum of Science, but also in Boston. Ladies and gentlemen, Feng Zhang.
Thank you very much.